Well, good morning or afternoon or evening, or in my case, midnight. And welcome everybody to the 6th session of the Global Ocean Oxygen Network, or GO2NE webinar series. I am Jodie Rummer. I am based at James Cook University in Townsville, Australia, and I will be moderating today's session. Today, here in Australia, and more specifically where I am based, it's important that we acknowledge the Bindal and the Wulguru Kabah peoples as the traditional owners of the lands and the waters where we live and where we work, and honor their elders, past, present, and emerging. The whole GO2NE team, which is part of the UNESCO Intergovernmental Oceanographic Commission, or the IOC, is very happy that you're joining us and our incredible speakers this morning, afternoon, or evening for global deoxygenation across scales from the tiniest to the largest in the oceans. But before we get started, please join me in congratulating one of our members, Professor Nancy Rabelais, who was just elected into the National Academy of Sciences. This is a huge honor and greatly deserved. Congratulations, Nancy. We're very proud of you. Now, before we bring in our speakers, if you are interested and want to speak at one of our upcoming webinars, please submit a short abstract. The link is also going to be shown in the chat box. We anticipate that our next webinar will be held on the 26th of May. And next, a bit of housekeeping. You as an attendee have the option to type your questions in the question box, and I will moderate them after each speaker. The chat function can be used throughout the entire webinar to share additional information, or if you have any logistical problems, we'll be monitoring that throughout. So without further ado, we have two great scientists speaking today. We will begin with Sarah Wolf, who will present for 20 minutes, followed by a 10-minute question and answer session. Likewise, David Sims will join us for 20 minutes, followed by 10 minutes for question and answers, and then we will conclude. So first, I'd like to invite Sarah to share her screen. All right, are we able to share your screen, Sarah?
I think so. Does it look all right?
Perfect. Okay, Sarah Wolf is a PhD candidate in the Department of Microbiology at Oregon State University in the US, and she is co-advised by Dr. Francis Chan and Dr. Stephen Giovannani. I first learned of Sarah's work because of her immense capacity to communicate her science in a clever and accessible manner, and I was convinced immediately that she'd be a really fantastic addition to our webinar series. Sarah, I'm super excited to hear you discuss your research on what microbes think about our ocean's deoxygenation problems. Welcome to the webinar, Sarah. You have our attention.
Great, thank you, Jodie, for that introduction. So welcome everyone. It's good morning. It's about 7:04 here in Oregon. My name is Sarah Wolf. I'm a third-year PhD candidate in the Department of Microbiology at Oregon State University in Corvallis, Oregon. I'm co-advised by Dr. Francis Chan and Dr. Stephen Giovannoni. And so today you'll be hearing about microorganisms and sharks. So I'm going to be telling you a bit about the bacteria and how they are a driving force in our oceans. So today, first, I'm going to discuss the research and the science, so how we investigate microbial responses to coastal oxygen dynamics. And secondly, I was asked to share some of the science outreach I've been doing this past year, so I will share that with you towards the end of the talk. So what happens to the ocean oxygen content when the planet warms? So there's this direct thermal effect because the solubility of oxygen is just lower in warmer water. That's chemistry, and this direct thermal effect only accounts for about 15% of ocean oxygen decrease globally. The other types of effects are indirect, so stratification, decreased mixing, and so on. And so these changes due to ocean deoxygenation have started to alter the balance of marine life, favoring these hypoxia-tolerant species at the expense of hypoxia-sensitive ones. So I want to talk to you about the eastern boundary upwelling system. So these eastern boundary upwelling systems, we call them EBUSs, comprise 1% of the area of the ocean. So you can see them circled here. While they only make up a pretty small portion of our planet, the productivity in this ecosystem supports about 1/5 of the world's ocean wild fish harvest. So these coastal ecosystems support the marine food web, which maintains healthy biodiversity, but also these areas support a plethora of ecosystem services. So I live here in Oregon, and so the work we do is off the northern coast in the California current system. And throughout the presentation, I've placed these QR codes so you can access any of the articles or links that I reference as I go. So what does this water washing up on the Oregon coast look like? I'd like to just share this short animation of how the water makes its way across the Pacific to us.
And there are actually two branches. One of them comes across the northern Gulf of Alaska. Another one takes this amazingly convoluted route. Along that path, the oxygen gets lower, the CO2 goes up, the pH goes down, it gets more acidic before it reaches Oregon shores. So if you think about the rising CO2 levels in the atmosphere, the water we're seeing at depth off Oregon has the signature of many decades ago.
Okay. So this water in the OMZ that comes across the North Pacific is already low in oxygen, and that's taken decades to get here. But once it hits our eastern boundary upwelling system, this low oxygen, acidified, nutrient-rich water gets upwelled onto the continental shelf, which causes this phytoplankton bloom, which results in increased bacterial respiration. And using up that oxygen in this area creates this secondary oxygen minimum zone on the shelf. And so this water that takes decades to get here, decades to draw down this oxygen, we see that the bacteria can create these secondary OMCs in just a matter of weeks. And so what I study is sort of the last mile of this process. The evolution of these secondary OMZs leaves our coastal communities really vulnerable. So our communities have a great interest in maintaining our fisheries and biodiversity. It's a great source of economic gain here in Oregon, as you can see. Here's a quote from a guy who's been a crabber in Oregon for 40 years. And he as hypoxic water can show up as a flip of a switch. If there's crabs in the pot, they're just dead. So he's finding dead crabs in his crab pot. And on the Oregon coast, we didn't actually see this phenomena until 2002 of seasonal hypoxia. But every summer after 2002, we've seen hypoxia to varying degrees. And with future projections of increased oxygen loss, this is a significant problem for our communities. So I'd like to revisit this idea of bacterial respiration drawing down oxygen in this secondary OMZ. So we have light and nutrients. We have an influx of nutrients coming up onto the shelf, and phytoplankton are just marine plants. So if you give them light and fertilizer, essentially, they're going to grow. So you have this phytoplankton bloom, and as the bloom senesces, the bloom, the cells begin to die, bacteria begin to decompose those that detritus. So there's more phytoplankton means more decomposition. So as they die, the bacteria start to consume this phytoplankton, which decreases the amount of oxygen in the water column due to increased respiration. And so organisms in the ocean need oxygen to survive. So this depletion can take a real toll on organisms, especially benthic organisms. So here's a photo of a Dungeness crab die-off that we found in Oregon due to hypoxia. So you can see why this would be a really big problem for our crabbing communities. So this is that part of the process where I investigate this last mile of this dissolved organic matter in the ocean. So let's talk about the microbes. So each drop of seawater contains 1 million bacterial cells. So if the ocean covers about 70% of the planet, the ocean is huge, and this exists in every single drop. Well, this is an artist rendition, but essentially, there's 1 million bacterial cells in each drop of the ocean. So while these microorganisms are extremely small, they're hugely important for biogeochemical cycling. So within that drop of seawater, the microbes in the ocean are extremely diverse. So they're the base of the food web. They cycle nutrients in the ocean. They sort of ping pong carbon, through the ocean. And here's a fun figure that is a comparison of size range of those phytoplankton relative to macroscopic objects. So when you look through a microscope, the difference in size between the phytoplankton is very significant. It's actually, this is a micromolar scale and this is a, macroscopic. And so if you think about tossing a fish, a single fish into the city of Manhattan, that's like a giant size difference. And that, if you think about Prochlorococcus in comparison to Trichodesmium, that's also a very big size difference. And that's what's really so amazing about the marine microbial world. So as oxygen decreases in the water column, we see that energy is shifted from pelagic macrofauna, such as mobile predators or sharks, which David will be talking about. And on this figure, we see that the axes are inverted. So on the left, we have energy to mobile predators, and on the right, we have energy to microorganisms, and we have our oxygen concentrations here. And so we can see that as oxygen levels deplete, we see that the energy is shifted from macroorganisms to microorganisms, where in the absence of oxygen, microbes can use nitrate and manganese, iron, sulfur, and lastly, carbon dioxide as terminal electron acceptors. And so this has the ability to decouple predator-prey interactions and really changes the trophic exchanges that occur in existing food webs. And the shift has huge ramifications for the global nitrogen cycle, the global sulfur cycle, causing a net loss of bioavailable nitrogen in the system. We need nitrogen to make proteins, amino acids, and then when microbes begin using sulfur, they can create that toxic compound. It smells like rotten eggs, so they can create very toxic compounds. So my undergraduate degree is in microbiology, and in classic microbiology literature and methods, I often find myself thinking about oxygen requirements of microorganisms as a dichotomy. So we study microbes in the lab at a fully oxygenated bench, or we use these anaerobic chambers where we can stick our arms in and study microbes in the absence of oxygen. So if we think about, and we use words like strict or obligate to describe their oxygen requirements. And if we think about the microbes living in these dynamic, changing, hypoxic coastal systems, it's really required a significant shift of perspective for me to begin, what's occurring between the obligate anaerobe and the obligate aerobe stage. And the data suggests it's not binary. So this is some data from our OMZ, actually just from this past Saturday. It's a depth profile, and we see that the blue lines are oxygen concentrations. So as we go deeper into the water, the oxygen concentration falls lower and lower. But we don't see, strict anaerobe anoxia. So we see that it drops down to maybe 5 or 10 micromolar, but we don't see it going down to complete anoxia. So what's the deal? Why aren't these bacteria running that last mile, consuming all of that available oxygen in the water column? So what prevents them from drawing down all of the oxygen? And this is a network of interactions showing correlations of bacterial taxa in different oxygen minimum zones. So the size of the dots is the oxygen concentration, and the color of the dots are major taxonomic groups. Network figures can be sort of overwhelming, but this one pretty, clearly shows that as we go from oxic waters to suboxic down to anoxic, there's a significant transformation of the microbial community. So in oxic waters, the other bacteria, meaning, a very diverse community of bacteria, are dominant. And then as we go lower and lower in oxygen concentrations, we see that these major groups of taxa start to dominate the water column. So I'd like to highlight 2 groups of major taxa, one being the SEPO5 group, which I'll share about in a moment, and the other SAR11, which is beloved and extensively studied in our lab because it was discovered in the Giovannoni lab. It's the most abundant organism on the planet, and subclades of SAR11 are, they're the orange dots. So they're present in suboxic waters and some oxic, but they're, can be dominant in some oxygen minimum zones as well. So I just want to share this example of how remarkable these tiny cells are, that, some of these cells can change their morphology, so they can actually change their cell structures and their microbial metabolism across redox gradients. So organisms in the SEPO5 clade cross oxic and anoxic boundaries. And where we historically think of, organisms being aerobes or anaerobes and adapting to either oxic or anoxic conditions, SEPO5 really tells us a different story. So SEPO5 actually has the ability to retain sulfur in sulfur globules within its cell that it can use as an energy source when exogenous sulfur is unavailable. So this is an animation from Bob Morris's lab showing storage of these sulfur globules in SEPO5. And these cells are like the people that bring in food and sneak food into the movie theater. So they kind of hold on to that sulfur for later when they're going to need it. So I want to move on to how we actually investigate microbial function in our laboratory across variable oxygen concentrations. And we ask questions like, will microbial activity buffer or accelerate global ocean deoxygenation? So this is our 16 degrees Celsius chamber here at OSU, where I've lived and spent the last year or so of my PhD designing and fine-tuning this system. We call it our data machine because it's a mesocosm system that allows me to actually recreate OMZs in the lab. And these experiments run for about 3 months' time when I do an experiment. And I'm launching one very shortly. So it's a lot of work, but it's super exciting because we get so much data out of it. So what do these experiments look like? First, I collect seawater off the Oregon coast out of the Hatfield Marine Science Center in Newport, Oregon. And then I bring back about 200 liters of seawater and we spike that water with nutrients and we create a phytoplankton bloom. And the other grads joke it's like a club in the 16C because it looks pretty cool. But this was a bloom that we formed in the late summer, so it only took 3 days to peak. But in the wintertime, sometimes these blooms take a week or so to do. And these are just a picture from the microscope of some of these really cute chain-forming diatoms that we have present in our blooms. So from there, the system is set up to have 2 separate treatments. So a saturated treatment and a hypoxic treatment. And we've engineered the system so we can actually mix our own gas, which allows us to change the oxygen concentrations to test various hypotheses. If we want to do microbial physiology experiments and have microbes in culture in the lab where we can really dig into the mechanisms, we have to have some way to control the environment. And if we're just sampling, every summer and there's varying degrees of hypoxia, our system really allows us to be consistent and test really, biochemical hypotheses. And so from these carboys, we can sample water consistently throughout the experiment. And we monitor things such as bacterial community composition. So who's there, the respiration rate, gene expression, like what they're doing, what do they have the potential to do, the total dissolved organic carbon. And then we've moved on even now, other folks in the lab who study vitamin B1 and arsenic compounds in the ocean, they're also using my data machine to test hypotheses about those topics. So how can laboratory, microbial physiology studies help us combat ocean deoxygenation practically? So we're using my data machine to target and identify potentially previously unknown or overlooked fundamental cellular mechanisms. And then from there, we can take these studies and we can take our hypotheses to the field where we can determine if the phenomena that we're witnessing in lab are actually seen in our Oregon coast OMZ out in nature. And then from this, we hope to be able to improve biological variables in global climate models because oxygen dynamics in eastern boundary upwelling systems are, the product of really complex biogeochemical and physical factors. And so changes in the ocean on our coast are really hard to predict. And so the impact of ocean deoxygenation, we know, won't be evenly felt across ecosystems. And so the Oregon coast is actually classified as an early impact system, meaning that we're going to get hit sooner. And from our research, we hope that we can increase this body of knowledge so that other ecosystems that can learn from our experience by these modeling variables. So shifting gears, I was asked to speak about my science outreach efforts. And so I want to share with you sort of how I've adapted this during the pandemic. So science outreach and science policy communication are major hobbies of mine, and I just love to share how cool and really important microbes are. So last spring, when the pandemic hit, we all know microbiology becomes a household hot topic, and we transitioned from in-person life to remote work and school. And a lot of my friends and family, who are parents, were really struggling with this transition with their kids. And I'm a first-generation college student, so I come from a very large family. This is just my dad and his first cousins, our wolf pack in 2019. But I sort of felt uniquely positioned when I thought about how my family and friends were struggling to keep their kids engaged with anything at home, especially science. And so at that point, it really wasn't clear whether school would be remote long-term. A lot of schools suspended lessons because we thought, maybe it'd just be a few weeks or a month or so. And so I started thinking like, okay, do these people know that microbes are really important in the environment and they're not all bad and pathogenic like COVID? And like, do they really even understand what I study, and how can I, think about translating that to them? And so I began brainstorming how I could use my connections and my skills to engage my network who don't have regular exposure to research science specifically. And so in my life prior to grad school, I worked in pediatric dentistry and I nannied for a while. And so I kind of felt like I wanted to make sure that the kids that I know and love at home, were thinking about science, and they felt like they had something to be enthusiastic about. Then Zoom became such a part of our world, and I thought maybe if I teach the kids something, then they can cheer their parents up with some fun facts. And so I began Zoom lessons. And I first started teaching kids about taxonomy and the naming scheme of organisms. And it was really fun to see them take their yogurt containers and see that lactobacillus in the ingredients list was actually a rod-shaped bacteria. And so from there, we moved on to, other topics like astrobiology, ocean deoxygenation, and so on. And so through this, I was able to try to, use my platform as a graduate student and a climate science trainee to increase access to research science and attempt to demystify climate science. So these Zoom lessons took off really fast. I was doing them literally every day of last spring, but it was a super fun part of my day. And now, that really pushed me to create a website where I could point parents to resources rather than sending out a ton of emails. And so now I do a lot of guest lectures for K-12 classes. And on our website, or on my website, I have different lessons. So our lab created a board game called Oligotrophic, which simulates how biomass moves through the ocean. And it can be downloaded for free there for teachers to use. There's a full lesson plan and data, sheets, everything there. And I'm also very close and excited to be launching a virtual mystery hypoxia online simulation, where students will try to figure out like, why are crabs dying on our coast? How do I form hypotheses and use the technology available to solve deoxygenation problems? And then in the last few months, I've got really into STEM children's books. So thanks to our Corvallis Public Library, they have a great selection. And so I'll be hosting 31 days of STEM books on my Instagram in the month of May, starting Saturday, where I'll share a new STEM book every day. So overall, I just feel really blessed to work for Steve and Francis, who have fostered a very integrative approach to my graduate training, where I feel supported and that these skills in like science communication, outreach, and policy are valuable and encouraged. And this approach allows me to feel challenged to think outside the box, to do use-inspired basic research science. And then I have opportunities to be involved in local policy. I actually started my undergrad as a political science major, and now I'm serving as a advisor on the Environmental and Natural Resource Committee in my county here in Oregon. Plus, our lab creates curriculum and trains Oregon K-12 educators to give lessons on cutting-edge research science through our SMILE program. And just all of this has made my academic journey really enjoyable and filled me with purpose, like especially during this challenging year. So with that, I'd like to acknowledge both of my labs, the GO2NE lab and the Chan lab, our funding agencies who've supported this project, including the Oregon State University College of Science and the Simons Foundation International. This is a photo of our lab. Pre-COVID, we took these pictures for a Christmas card. Also, I'd just like to acknowledge the really cool and resilient people on the North Coast. I grew up on the North Coast, the fishing communities, and those experiences really keep me positive about what I'm doing and the important mission that we've been tasked with. Thanks for your attention, and now I will take questions.
Thank you so much, Sarah. That was fantastic. Really, really interesting talk. And I'm already seeing that we have 4 quite detailed questions that I'll start fielding. So our first question is from Andreas. And Andreas is asking, why is there a maximum in energy to mobile predators, minimum in energy to microorganisms at about 160 micromolar oxygen?
So I think they're referring to this blip on the about right here. And so I think that, you know, different organisms have different requirements for oxygen. And so I personally don't study a lot of the macro things. And so I think this has to do with the physiology of macroorganisms.
Okay. So yeah, as we're saying, if you do have a question, please use the question box, and I'm happy to field these questions for Sarah. Our second question is from Montserrat, and we said, you showed a CTDO profile. The oxygen sensor of CTDO is not good to measure anoxia because it never reaches anoxia. The sensitivity is not good. Other sensors need to be used to say that it is not oxygen in the water column off Oregon. So maybe you can comment on how oxygen is being measured.
Yeah, so in addition to CTDs, we also do respiration experiments on our cruises. And so that was a 5-day research cruise where they were also doing respiration experiments with optodes.
Okay, so it sounds like a mechanistic or a technique question there. All right, so our third question, I'm probably not going to get this name correct, Melchizedek. Very interesting, thank you, Sarah. My questions: one, does the oxygenation phenomenon only occur in the surface layers of the ocean or also the bottom? And two, is there a latitudinal variation, for example, between the temperate and tropical regions? Thank you.
So where we're at in Oregon, we have deoxygenation, you know, deeper. We have like kind of in that video, it talked about a deep acidified, you know, low oxygen current coming through the ocean. And so we do have it in the deeper depths, and then it gets washed up onto the shore. So it does kind of change and move up in the water column. And then I think the second question was about like how deep OMZs are across the earth. Is that?
So between temperate and tropical regions.
Got it. You know, that is I have not studied much about the, you know, the more maybe the Humboldt current system. Most of my focus is in the California current system, so I could get back to you on that. And I'm sure there's some research online that can point you to different OMZs.
Excellent. We have a question from Victoria. How is pH affected by deoxygenation?
So we actually, here in Oregon, we have a Ocean Acidification and Hypoxia Council. And so these things they kind of go hand in hand, especially here on the Oregon coast. So they're definitely involved.
Wow, the questions are just coming in. So we'll try to get through these before we have to move to our next speaker. Patrick is asking, could you give some introduction of S protein in SUP05 and what the function is?
So that organism, I could not give you the detailed information about the types of proteins in that organism. I personally don't study SUP05 metabolism. We study more of the SAR11 group, but that was just a really cool animation and kind of an example. But I do have some papers. If you drop your email in the chat, then I would be happy to send over some more information about SUP05.
Excellent. And Montserrat is saying thank you because you must have answered that question perfectly. Mats is asking, will it be possible to look at the presentation after this session? Yes, they will be. They are being recorded, so we will post presentations after the session concludes. Another question from Crystal. At the Oregon slope and continental shelf, what is the respective role of microbes in the water column and sediment in consuming oxygen?
So on the continental shelf, the microbes, they play a very large role because so we have this already low oxygen water being washed up, and where that phytoplankton bloom forms on the surface layer, as it sinks down, that's where a lot of that microbial activity is occurring. And so the area on that continental shelf is like booming with microbial activity.
Excellent. We have comments of very good presentation. Thank you very much. Brendan Tarley, you showed CD profiles and asked why the oxygen doesn't go to 0. So why don't we see strict anaerobes in the OMZ, or why doesn't the OMZ go anoxic?
That is actually what my PhD thesis is testing. So I'm currently working on various hypotheses, trying to figure that out. And so, you know, we're kind of eliminating variables that we could, you know, eliminating variables in our OMZ in the lab that we can really hone in on and test hypotheses why this is occurring. And so I'm currently working on trying to figure that out.
I think a similar question is coming from Yushin. Do you think the OMZ in Oregon is under escalating because the OMZs are developing in different directions? Some are becoming severe, some are relieved?
So our OMZ is typically hypoxic during the summer. So hypoxic, I think, is 63 micromolar is the standard, you know, threshold. And so we see our waters go hypoxic, occasionally suboxic, but we don't see complete anoxia in Oregon.
Right. So Mark Costello asks, are the anaerobic bacteria more primitive than aerobic, reflecting Earth's evolution?
I know, you know, if we think about our low oxygen shallow seas back, you know, I think in like the Precambrian, then some of those organisms probably have evolved from that type of function, surviving and having to live in that type of ocean. And so some of the organisms that may be in the deep sea, the anaerobic ones, I'm sure have evolutionary lineages back to, you know,
Yeah,
older.
Great question. David Kirschman is mentioning great talk, and a devil's advocate question, which I get a lot. David says, how does information about bacteria community composition help us understand ocean deoxygenation?
Oh, that is a very good question. I think, you know, the community composition level, the 16S, you know, our RNA data that we can get from these experiments, they can kind of give us just a peek into who's there, but really that gives us the access to, in the Giovannoni lab, we love to culture things. And so that kind of gives us access to see if there's a dominant player that we can culture in the lab, then we can really test hypotheses more in detail. But, you know, the 16S data can only provide us with so much, but it kind of gives us an insight so that we can maybe do metatranscriptomics later on and test more hypotheses in depth about specific cell types.
Excellent. A big thanks from Patrick Chen. Sachiko says, very interesting presentation. Thank you very much. Sarah, could you talk about the detail about involvement in local community beyond academia, because deoxygenation may be unknown to citizens? And that's something I was super interested in as well, Sarah.
Yeah, it's interesting because it is an issue, like, you know, it's a very prominent issue that we have to face at a much faster rate than other problems, maybe globally. And a lot of people don't know about it per se. And so just through, I'm really involved in I'm a parishioner here at St. Mary's Catholic Church in Corvallis, Oregon, and I'm part of a climate group there. And so just communicating these issues in that community, and there's interfaith climate groups that we work with. And then also just finding local opportunities. Like I found this opportunity to be an advisor on my local county where we advise the Board of Commissioners, you know, because we do research science. And so just being able to interact and talk about the work that we do in a way that they can understand is really beneficial, because as soon as they find out about ocean deoxygenation, they're like, whoa, I had no idea. Like this is happening just, you know, an hour away, because the coast is really close to us. So I think just talking about it and sharing with them, and they're super excited.
No, that's great. I think that's a super important part of this whole outreach aspect. You know, we're sort of preaching to the choir, so to speak, amongst ourselves, but, you know, getting out there to other communities and other groups. One last question I think that we have from our Elaine. Did you quantify the microbial diversity in suboxic conditions compared to anoxic conditions?
So this summer, we just wrote a proposal for an undergrad to work with me in the lab where we'll be doing geometric series of oxygen concentrations, because right now with the data machine that we have, we test 2 oxygen concentrations and then we can kind of fluctuate them. But we're sort of honing in on that question of like just not 12 micromolar or 260, like what's the in between. And so that's what we're working on this summer. And we'll be doing bacterial community composition on that.
All right, well, we need to leave it at that. Unfortunately, and we've got more questions coming in for you, Sarah, which is not surprising. Such a great seminar. Thank you so much for joining us. We're going to switch the screen over to David.
Mm-hmm.
And let's see.
I think I just gave him permission.
Allow David to share his screen. Perfect. So thank you so much, David, for joining us. David Sims is a senior research fellow at the Marine Biological Association Laboratory at Plymouth in the UK and a professor of marine ecology at the National Oceanography Centre at the University of Southampton, also in the UK. I've been following David's work for some time, but what really prompted me to think of him for this webinar series and to discuss how some of the largest ocean life tackles global deoxygenation is a paper he recently published with several colleagues titled Climate-Driven Deoxygenation Elevates Fishing Vulnerability for the Ocean's Widest Ranging Shark, which was published in eLife earlier this year. So we'll shift from the tiniest to some of the largest ocean life. And David, thank you so much for joining us. You have our attention.
Okay, can everyone hear me? Is that fine? Jodie, can you hear me?
I can hear you. You sound great.
Great, fantastic. Just testing the technology there. Okay, thanks very much, Jodie. Thanks for the invite. It's great to be here and to talk to you about ocean deoxygenation and pelagic sharks. I guess before I should get into the talk, I just need to thank the people that you can see in front of me there. That's the research team collaborating with Cibio at the University of Porto. And that's what research groups look like these days, everyone in their sort of video boxes. But I'm going to talk about Nuno's work, Marisa, and Bruno and Gonzalo in particular. So thanks very much for providing such great science for me to talk about. And when I say I mean we, and when I say we, I mean they. So just to get that clear from the start. Okay, brilliant. Okay, thanks. So yes, an important change is occurring in the ocean, global open ocean deoxygenation. And it's been estimated that about sort of 2% of the global ocean oxygen inventory has been lost since about 1960. And as Sarah actually very ably described, the sorts of declines we're seeing are due to ocean warming, increased stratification, changing deep water circulation and ventilation of areas, and also increased microbial respiration, as she alluded to. So there's big changes going on. And obviously, a deoxygenating ocean is likely to be a particular problem for these high oxygen demand predators, such as the short-fin mako shark, which can swim along at burst speeds of 70 kilometers per hour, about 40 miles per hour or 20 meters per second, if you want it that way. Very fast, they'll be using probably at those sorts of speeds upwards of sort of 500 milligrams of oxygen per kilogram per hour. And it's likely that as we see oxygen decline in certain areas, it's going to cause shifts in the distribution and behavior of these animals. Also, here's the blue shark. It's likely that this is the world's widest ranging shark. It moves across all of the oceans through a broad range of habitats and is likely to encounter these low oxygen environments. Understanding environmental effects on pelagic sharks in particular, these oceanic wanderers, is really important because they've been declining over the past, at least the past half century. About a 70% decline in abundance of about 18 species of oceanic sharks and rays have been recorded. And for the short fin maker and the blue shark that I mentioned, that's changes of about 30 to 40% reduction in their abundance. And that change has been attributed to overfishing principally. And sharks, such as the blue shark, are not only the sort of target of longline fisheries, as you see here, but they've also for a long time been bycatch of other target species, such as the swordfish you can see here on the left. So sharks are being taken by longliners, by purse seiners, and also set net operations as well. But it's those large industrialized longlining vessels that I'll focus on in this talk, because over the last sort of 70 years, they've increased, expanded across the global ocean. So their reach has gone into areas that formerly weren't exploited, and where sharks had generally a refuge from exploitation. So because there is movements of sharks across the ocean and there is this extended reach of fisheries, we have to take a global view of where the sharks are in relation to these fisheries and consequently to the changing environments that we see them occupying. About 5 years ago, myself and Nuno Queiroz developed this or initiated this global shark movement project, which is essentially bringing together about 150 researchers across 26 different countries to pool satellite tracking data of pelagic sharks. Using this sort of technology where we follow individual shark movements, but we also use devices that can record aspects of their movements and behavior to give us a much richer picture of what the sharks are doing. And one of our sort of first studies that we did was to analyze the pelagic shark movements in relation to fishing vessels. And one thing we found was that here's our shark density here at the top from our satellite tracking locations, overlaid with our fishing effort data. So this is longline fishing data from the Global Fishing Watch. And when we overlay those two and drill down to look to see where the overlaps are and the intensity of the overlaps, we found that about a quarter of the space use of sharks per month were overlapped by these longliners. And that can even be up to 76% for the blue shark that I showed at the start, which is up here. So there's big overlap and also intensity, fishing intensity in these habitats that sharks prefer to occupy. And one in particular was a shark space use and fishing overlap hotspot was off the coast of West Africa. And we became interested in this in particular because it is also an area where there is an oxygen minimum zone in the mesopelagic from about 200 to 800 meters deep. And this has been studied quite intensively by researchers in Europe and elsewhere, and was the subject of, in part, a study in 2008, which looked at the expanding oxygen minimum zones. It was thought from about 1960, it was estimated that these oxygen minimum zones were expanding. So the volume of that low oxygen water was increasing. And it was proposed that this would reduce the available habitat for tropical pelagic fishes. And in the intervening time, over the last 10 years, there's been sort of anecdotal information from sharks, pelagic sharks, that that some species tend to avoid these areas. But we started working in this area to try and understand a bit more and try and quantify the responses. And so, caught in the middle, ocean deoxygenation and longline fishing effects. In the time I've got left, I just want to really sort of try to answer three questions. Does that eastern tropical Atlantic oxygen minimum zone affect shark depths? What are the potential drivers of any changes that we see? And are these sorts of changes, if they happen, likely to change or affect the vulnerability of sharks to longline fisheries? And very much we focused on trying to quantify the shifts in movements and behavior of the sharks, of the fishers too, but also the catches that were being made in these regions. So we started, as many others had before us, thinking about pelagic fishes, thinking about expanding oxygen minimum zones, this increasing hypoxia, leading to habitat compression. And where there's habitat compression, there's a reduced vertical niche, which may attract more surface longlining vessels. Each of these vessels can deploy a long line about 100 kilometers long with about 1,200 baited hooks. And this can be done every day. And there are thousands of these vessels plying the oceans over the annual cycle. Obviously, it could be that some of the sharks that we're investigating aren't necessarily sensitive to at least low or moderate hypoxia. And so it could be that over the top of these oxygen minimum zones, we don't see that there's any particular vulnerability of the sharks to fishing. They're with perhaps behavioral or physiological adaptations that allow them to occupy these lower oxygen levels. Perhaps not the core of the oxygen minimum zone, but certainly the outer sort of shells, the outer sort of layers of it. But also we are interested in this idea of a habitat trap. If indeed sharks were restricted in their vertical movements and necessarily were aggregated by these strong boundary edge oxyclines of the oxygen minimum zone, and also if their prey was hypoxia intolerant, they would also be habitat compressed. And would this lead to a habitat trap where the sharks spend more time in this habitat compressed area than they necessarily would do otherwise if there wasn't that habitat compression of prey? So we started to investigate these hypotheses. And with Nino and others, we attached tags to the fins of sharks, blue sharks and mako sharks, in this case the blue shark, around the Azores with Pedro Afonso at the University of the Azores, and also in other locations in the Central Atlantic. And we looked at where the sharks moved to and how much time they spent in particular areas. But also with these tags that could record swimming depth and temperature, we looked at how much the distribution, the vertical distribution, was reduced by the presence of this oxygen minimum zone. We found that the blue sharks would seasonally move south. Many of them would move south. Some of the larger individuals would move south and occupy areas. About 65% of sharks spent more time there. They preferred that habitat over the oxygen minimum zone than elsewhere. And as you can see here, both blues and mako sharks are really not only sharks of the surface, our oceanic surface, but they're also sharks of the twilight zone. And you can see here this blue shark moving happily on a sort of daily or twice every other day basis, moving through the twilight zone into and down to waters maybe as deep as 1,500 meters, 1.5 kilometers down. The mako shark also in normoxic waters, moving down to as deep as 1,500 meters. So these sharks are utilizing the whole of the sort of Atlantic water column down to about 2,000 meters. The deepest dive I think we had was about 1,800 meters. So they're occupying that top 2 kilometer zone, and within that they spend most of their time at the surface. So clearly our mesopelagic oxygen minimum zones are something that the sharks in their daily activities would encounter. What we find is that actually here's a blue shark that is moving south and encountering the oxygen minimum zone off of West Africa, that eastern tropical oxygen minimum zone. And we can see that just by looking at that broad sort of dive depth data, we see that there is a shoaling of the maximum dive depth as that oxygen minimum zone is encountered. And one thing to say here, of course, is that we're using modeled oxygen. So, of course, where the oxygen is, where the sharks are, may be different to the modeled oxygen. And so we're, and I'll come on to my last few slides, will show some of our sort of current and future work that we're doing with developing tags. But we can see that generally here there's a as a response of these blue sharks to the oxygen minimum zones. This is different individual, again, a shoaling of those deeper dives. And indeed, that's what we see. It's not that necessarily there's a whole shift towards the surface. It's just that those deeper dives seem to be truncated. Here you can see that the time at depth, almost 100% of time at depth is spent less than 500 meters. In fact, about 91% of time above this oxygen minimum zone is spent in the top 250 meters of water. But they do maximum, there were dives down into deeper water, even where the modeled oxygen was quite low. So it's possible that the blue shark, as an ectothermic shark, lower metabolic rate than our sort of high performance short fin mako sharks, it's possible that they may make short forays into that oxygen minimum zone. So we put our dive data together with the environmental data to try to sort of describe and quantify responses with respect to environment, to try and appreciate what the drivers were. And it certainly seemed to be that there was blue shark habitat compression due to this oxygen minimum zone, but it wasn't just oxygen that was important. Here you can see what the general sort of trends is that dive depth shoals, or the maximum dive depth shoals, as oxygen at depth at 100 meters gets lower. And what we see with maximum dive in relation to sea surface temperature is that maximum dive depth increases as sea surface temperature increases. So over these oxygen minimum zones, there's strong stratification, often high sea surface temperature. And so we see the shark move a bit deeper. But as it gets very warm here, we see that again, that maximum dive depth shoals. And so there's very interesting relationships between the diving behavior of the shark in relation to oxygen and sea surface temperature. We also found that net primary productivity, this sort of biomass of phytoplankton, proxy, also was significant in our model. In other words, maximum dive depth, as you can see here in this plot, as the net primary productivity increased, we saw our maximum dive depth shoal. And that points towards, it's sort of circumstantial evidence in a way, it points towards there being more productivity in that sort of top layer. And that could be related to more foraging opportunities for fishes, including sharks. So our model pointed towards the habitat compression of blue sharks with this oxygen minimum zone. And next we wanted to see that question about vulnerability. Is the vulnerability of the sharks with that sort of a shift of maximum dive depth towards shallower water, is that linked to higher catch rates? And what we can see here are the movements and fishing effort of two of the most important fleets in the Atlantic, the Spanish and the Portuguese longlining fleets. And this is fishing effort, so where there's hot colors, that's more fishing effort. And you can just see here, we put a dotted line around the 3.5 milliliters of oxygen or 100 meters oxycline. And you can see that these longline vessels, if you look at the Atlantic as a whole, they're not only, they're going for this area where the Gulf Stream converges with the Labrador Current, a very productive zone where they've been exploiting sharks and other large pelagic fishes for a long time. But we can also see that along this northern boundary in particular of the eastern tropical Atlantic oxygen minimum zone, we see that there are hotspots of fishing effort. And indeed, we found that there was greater fishing intensity as well, which is the proportion of fishing locations in what are called area restricted search clusters. So the long lines that were being put down were being put down closer geographically by the same vessel, closer to where it deployed the line last, but also on consecutive days. So there was an intensive fishing effort in these hotspots. And what we find also is when we looked at the actual catches of the Spanish fleet, this is from the logbooks of the skippers of the vessels who actually were plying this area. We see that for blue sharks, there was a peak in catches as the DO at depth declines sharpest. So it seems that there are these boundary edge oxyclines that represent a threshold boundary for blue sharks, at least on average. And it may also act to aggregate them as well. And that could account for these peaks in catches that we're seeing along these sharp boundaries. And you see the peaks that in catches that we're also seeing here. So a habitat trap for sharks? Well, maybe it is. Maybe the sharks are not only staying in that area because of increased foraging opportunities, but they're also being aggregated by this oxygen minimum zone aggregative mechanism, these boundaries of oxygen through which to lower oxygen that they won't move into. So it does suggest the potential for overfishing of habitat compressed sharks. But as I mentioned before, my last few slides just relates to some of the work we're doing now and for the next sort of 5 years in our latest grant. And what we're attempting to do is to understand the oxygen where the sharks actually are. So we need to collect DO data where the sharks are, rather than perhaps relying entirely on the modelled oxygen. And Nuno Queiroz and Bruno Luero with Electric Blue at the University of Porto have developed this tag stemming from our studies on blue sharks and short fin makos, which will attach to the shark, as you can see there in this diagram, this beautiful diagram here, this sketch. You can see the tag attached. And the idea of this is that there's a DO sensor, there's also depth and temperature sensors, there's an accelerometer, so we can get the movements of the shark at very high frequency. We can start to look at tail beats of the shark as it dives and when it comes up. And then at a pre-programmed time, after 1 or 2 days, the tag releases, bobs to the surface and sends out a GPS signal of where it is, and we go and pick it up in the boat. Also in development is a satellite relay version of this, which summarizes the DO data where the shark, or through the habitats where the shark has gone. And just to give you a last couple of slides, just to give you a snapshot of where we are with this. This is some Azores fieldwork to test the devices. This is Ivo da Costa, a PhD student with us, who's tagging a beautiful big blue shark there. And that shark went off into the ocean, and then the tag popped off and was recovered successfully. And we can see that here. This was that blue shark. We're getting dive depth data, as well as temperature data and the oxygen data here. At good resolution as well for those 1 or 2 days that it was out. So we're developing, as part of this ERC project, this ERC grant, a novel oxygen sensing tag, which can give us all this sort of detail about the shark responses. And also to test this sort of habitat trap hypothesis, we're incorporating a very small video camera into the tag to see forward of where the shark is, to try and image the prey encounters and potential consumption, and to estimate prey abundance. How important are these oxygen minimum zone areas above and perhaps into the oxygen minimum zone for these pelagic sharks? We're also crazily, you know, you do these things when you write proposals, is to actually have to take direct measurements of shark forays into the oxygen minimum zone. And that's, we're going to develop a second version of this swim tunnel respirometer, this mega flume that Nick Payne and Jason Simmons developed in Australia. And where we're going with this, obviously, is to try and use this physiological and ecological effects on behavior to incorporate in better model estimates of fish habitat loss as a consequence of this expanding oxygen minimum zones and general sort of open ocean deoxygenation. So look, I'll leave it there. Thanks very much. And just to say that in the future, it could be that management measures will be necessary that specifically act to mitigate the effects of ocean deoxygenation on catch rates. In other words, considering climate change effects on fisheries and the management more so than we do now. So anyway, thanks very much, and I'll leave it there.
Thank you so much, David. That was a fascinating talk, talking about really emphasizing how all of these factors coming together can really influence how we're managing some of these ecosystems and these species for sure. We have some excellent questions for you. One from Benjamin. Great talk, David. Using your diving data, can you parse out if those deeper dives into hypoxia by the mako are from the surface, or are they from intermediate depths? Would it be possible that they go from hypoxic waters into colder, more oxygenated waters for replenishment?
Yeah, it is. I think you're right. It is possible that the sharks can sort of thread their way through this low oxygen and perhaps trade off the availability of higher oxygen levels, even within the outer layers of that oxygen minimum zone. And I think when we get to deploy those oxygen sensing tags, as we plan to do as part of that ERC advanced project, is to deploy them on sharks which are diving above and hopefully into the oxygen minimum zone. And I think we'll get a much better picture then on the strategies and the economy of the shark physiology. They're obviously making decisions about where to go and what to do, and it will be fascinating to know whether they actually can punch through the oxygen minimum zone, hold their breath and come out the other side, or utilize these sorts of threads of higher oxygen that might be in that oxygen minimum zone. So yeah, fascinating. I look forward, hopefully I can come back in 5 years with some answers, who knows.
No, that sounds great. And coming from someone who typically studies the more hypoxia tolerant sharks, most sharks are not that way. So sort of these holding our breath payback ideas are super interesting. We have a question from Melchizedek. Thank you, David. From your view, is there any remote chance that the pelagic sharks will gradually adapt to the minimum oxygen zone via natural selection, or do you feel they are doomed if the situation persists unchecked?
Yeah, that's it, isn't it? It's how fast the change happens and how fast the shift is in oxygen into habitats that they formerly occupied. Obviously, one could suggest that they've been adapting to low oxygen and high oxygen for millions of years. You know, the ones we're seeing now have probably they were probably evolved in their sort of current form about, you know, sort of 100 to 150 million years ago. Obviously their relatives go way back, you know, to 400 million years. So potentially, of course, they have adaptations, behavioral, physiological adaptations that allow them perhaps to, as we saw there were some dives that were really tantalizing, deep dives when we located them above the oxygen minimum zone by satellites. So where, you know, were they going through? Were they, you know, expressing those adaptations? We've got some work going on actually with Rui Rosa and Marisa Vedor, who I mentioned there, that starts to, and they've taken blood samples from sharks that in these or diving in these oxygen minimum zones and outside, and their results look to be fascinating. And I think when we start getting that information, we'll be much better off. We're in a much better, stronger position to ask, well, what are the adaptations these animals have, and how long have they had them?
Absolutely. Mark Costello is commenting on that, just wondering how long a shark can go anaerobic, and you know, of course, that's going to depend on the species. And yeah, I think, as David is suggesting, a very much watch this space sort of answer with that one. Let me move to George Cummings. Great intel on new tech applications, and now to see what we can learn with it. Let's see, we have another question. Could you say something more about how sharks and fishing fleets might respond to small scale and transient shoaling of the upper OMZ boundary associated with mesoscale variability? That would be fantastic. And that's from Ika Kern.
Yeah, so I think, I mean, the fishers will respond very quickly, as will the sharks. We know a bit more about the fishers perhaps at the moment than we do about the sharks, because we're tracking, you know, tens of sharks, but we're tracking thousands of fishing vessels. So that data I showed there was for thousands of longliners. And certainly when catches dwindle, so for example, if long lines are being laid, usually the baited hooks are about sort of 100, 150 meters depth, maybe up to 50 meters sometimes. They will adjust the hook depth depending on where they're catching. And that can be in response to low oxygen. And if catches dwindle, they either might change the depth or the fishers will move somewhere else. And you see very clear search patterns, just like we've analysed actually the fishes in the same way we have the sharks and published those papers, and they're fascinating. You know, how the movements of a vessel is essentially a super predator moving around. And they respond very quickly to changes in the environment. They have lots of information, satellite information about sea surface temperatures. They can lay lines across fronts, for example. The sharks are using exactly the same sorts of information. They have sensors for temperature and oxygen and will respond very rapidly. I mean, we've got probably temperature sensors on sharks go down to about 0.0001 degree C per centimeter. You know, so they're rapidly detecting changes in temperature. So yeah, rapid is the word I would use.
I guess the question is, who's the predator here? And that's kind of coming off of you know, is it the fishing vessel or is it the shark? And, you know, both are responding in changes in behavior. Christopher Johnson had asked about changes in behavior under low oxygen conditions. So yeah, I mean, I think we have a lot of questions with respect to ecophysiological changes, changes in behavior, a lot of comments on just what cool work this is. So I think you're going to have heaps of emails and a lot of questions stemming from this fantastic talk. Thank you so much.
Yeah,
David,
no
we're going to conclude there. Yeah, so that concludes our 6th GO2NE webinar. Thank you all so much for joining us. Keep an eye on our YouTube channel and our webpage as we will host this video in due time. And please do check out our fantastic speakers on their websites and on their social media channels as well. Drop them a line if you have further questions, and stay tuned for details regarding our webinar number 7 for late May. Have a great morning, afternoon, or evening. Thank you so much. Thank you.